Temperature effects on baby coral symbiosis. Imagine a coral reef like a major company. Coral animals are the CEOs, but they rely on a mutually beneficial relationship with microscopic algae. These algae live in coral tissues, providing them energy and their color. But when the ocean gets too hot, corals enter a stress response called bleaching, where they expel the algae. It's like losing your entire workforce. The reef begins to collapse, biodiversity drops, and nearby communities that depend on reef suffer the consequences. Because ocean temperatures are rising more intensely, coral recovery now depends on the survival and success of coral babies, the next generation. These babies have to correctly choose which algae from the surrounding environment is the best partner for survival. Some algae are more heat tolerant, while others have more nutritional benefits. Choosing the right partner is like a startup trying to find the best business alley during a market crisis. Those decisions can make or break survival. Past lab studies only looked at coral babies exposed to one algae species at a time, but in nature, coral babies encounter multiple algae types simultaneously, and these algae compete to enter the host, especially under heat stress. That brings us to the study's biological question. How does heat stress affect the ability for different algae to enter the coral and succeed among competing algae in coral babies when multiple options are available? Researchers hypothesized that at elevated temperatures, coral babies would partner more with the more heat-tolerant species than with the more heat-sensitive algal species, and that the increasing heat in the ocean would reduce the amount of coral babies and increase coral deaths over time.
Now let's look at how the scientists studied how heat and algae types affect baby corals and their symbiotic relationships. First, six adult corals were collected from the reef, and the scientists waited for these corals to release eggs and sperm. These were then mixed to create baby corals. The larvae were raised in clean water at normal ocean temperatures, which is about 27 degrees Celsius for over the span of 10 weeks. Then the scientists grew four different types of algae, as you can see the different colored bottles in the lab. The scientists tested how each algae grew inside the larvae at three different temperatures: 27 degrees Celsius, 30 degrees Celsius, and 31 degrees Celsius, as you can see the three things in the chart. For each temperature, larvae were placed inside jars with only one type of algae, a mix of all four types of algae, or no algae, which served as a negative control, which made sure the experiment went through properly. And each treatment was repeated 3 times to keep the result trustable. To do that, they first used a microscope. This is a tool that lets you see very small things with your eyes that you can't see in a regular way. They use it to visually check which larvae had taken in the algae. As you can see in the panel B, the larvae is glowing pink, and that pink glow comes from all the different algae inside the larvae. This gives scientists a very quick first look at whether the algae was being taken in at all. Next, they use the hemocytometer, which is a very small glass slide with a special grid on it, and the scientists basically put a droplet of cells on it, and then they count the number of cells within a certain number of squares using the microscope. This gave them precise numbers of how much algae was within each type of larvae. Finally, they use this tool called a ddPCR, which basically stands for Droplet Digital PCR, and this is a kind of a DNA test. It breaks a sample into thousands of tiny different droplets, and each droplet is then tested to see whether if the algae has DNA inside and how much of it. This is done because not all algae have the same amount of DNA, so before counting the number of cells, the scientists measured how much DNA was within each cell so they can get an equal count and better compare the cells of each algae type. All of this is basically done to see how much algae grew inside the larvae, comparing the algae between how it stood alone and versus in competition to figure out which algae basically won against the others and to just overall track the algae community change over time. Finally, they use computer programs to keep track of charts and other results.
The results of the researchers' first experiments were really looking to measure how many of the coral larvae actually wanted to bond to any of the four types of algae and if they had a preference to a certain kind. Much like people with our favorite foods, scientists had considered that maybe coral, likewise, had preferences for a certain type of algae. Measuring the results on days 3, 7, and 14 of baby coral at 27, 30, and 31 degrees Celsius, and finding the average amount of baby coral that had bonded to the algae at those temperatures and times, scientists found that in lower temperatures, the larvae preferred Cladocopium algae, a common algae, and in higher temps preferred Fugacium, a more rare algae. But at all temperatures, the baby coral were able to bind to some type of algae. In the second experiment, by introducing the larvae to a mix of all four types of algae, scientists believed that they could figure out which type of algae the larvae's true preference was. By setting the same experiment and timeline up as the first one, but this time only giving the baby coral a cocktail of all four algae, the scientists noted something interesting. Rather than just discovering a preference of the larvae, scientists found that at the lower temperatures of 27 and 30 degrees Celsius, the coral larvae actually wanted all four types of algae, with three out of the four types being found in almost equal amounts. But at the higher temperature of 31, the corals only wanted three types of algae and denied the fourth one almost entirely. So, how did they decide what the favorite algae of these corals were? Well, scientists had to take individual larvae from each test and measure out the average amount of each algae's cell types within the larva. This led to interesting results, and it showed in almost every single case the Garicladium, another rare type of algae, or Durusdinium, a common type of algae, were not only present in the larvae, but were predominant in their total algae cell counts almost every single time. Unfortunately for Fugacium, however, it dominated in only three single larvae that were measured and was consistently seen to be the least favorite out of all types of the coral. Ultimately, the experiment showed that preference of the larva was heavily reliant on the temperature they were at rather than the days or the actual type of algae.
Stress caused by heat reduces the diversity of the algae available to the coral larvae. That means that fewer types of algae can survive the higher temperatures that we used in this experiment. However, a few different types of algae were able to survive the environment at 31 degrees Celsius. Two of the types of algae, Geracladium and Fugacium, performed well at the high temperatures. This went against what the authors originally thought would happen, but that's a good thing. See, these algae act more like background actors in a natural environment. They're more rare when it comes to their ability to affect the coral larvae in oceans. Also, we saw that when all four of the algae were present, there was the highest amount of success of algae associating with the coral larvae. When there are more options, the best algae available can mingle with the coral larvae. This also happened quicker when there was more options, seeing as was more algae by day 3, when all four algae were present compared to only one algae being present. This is a representation of the real-life environment and relationship between coral larvae and algae because in the oceans, multiple types of algae are present all at once. Additionally, the scientists found that in all treatment cases, the coral larvae survival decreased dramatically as temperature increased. As temperature rose, some of the coral larvae populations decreased by up to 50%. At the highest temperature, some of the treatments resulted in none of the coral larvae surviving by the end of this experiment. This displays a stark reality about the effects of global warming on coral larvae populations. Even though the algae could access the coral larvae, that did not mean that the coral would survive. The algae is not the only factor that affects coral health. Ultimately, there are limits to optimizing heat tolerance of algae types. However, we agree with the authors of the paper in that introducing more heat-resistant and rare background algae can have more useful impacts at higher temperatures. As the oceans grow warmer, species that are more heat tolerant can provide a better ability to form symbiotic relationships with the coral larvae. We also agree that the algae diversity is crucial for coral larvae success at higher temperatures.
To conclude, this study reveals that the quiet, behind-the-scenes algae partners might be more valuable than we previously thought. In high-stress environments, they sometimes outperform the dominant algae like Cladocopium. Coral babies formed very different algae partners versus when the coral babies only had one option. The rare algae might offer key advantages under heat stress that are invisible in one-on-one experiments. So why does this matter? Coral reefs support entire economies, from fishing to tourism, and they also protect coastlines from storms. If these overlooked algae can help corals survive rising sea ocean temperatures, they could become a new tool in reef recovery and restoration planning. This could change how we approach it, treating the rare algae as a strategic asset. For future steps, researchers need to follow these symbiotic partnerships over time. Do these rare algae continue to benefit the coral as they grow into adults, or do the effects fade out? And how often do these same partners show up in the wild outside of the lab? By understanding where and when rare algae dominate naturally, we might find new ways to guide coral recovery.